So thanks to everyone for coming tonight. We are in our, do we have a December lecture?
Yes.
Second to last lecture for 2023. If you haven't grabbed one, please grab a flyer. It has our 2024 lecture schedule that is up on it with a little QR code for registration, so have a look at that. Tell your friends, tell your family, we're really trying to build these back again because I think it's so important, as you guys have heard me say many times, to do this kind of outreach for the community, but it's actually really boring to give a lecture up here and have nobody around. And so it is much more fun to have people in attendance and asking questions. And so please, tell everybody to come. Back in the day when we had a lot of people here all the time, I would give a big long introduction to the Stem Cell Research Center and California Institute for Regenerative Medicine and how important this is for scientific research. And show slides. I think we're still in the zone of being a little bit more casual for that, so I'm not going to go down that rabbit hole, other than to say it's been a really exciting time for the Stem Cell Research Center over the last few months in particular. So most of you know our Alpha Clinic is well underway with our rebooted CIRM funding. We actually have 30 clinical trials running at present. We're on track to have completed over 60 for this year, so super, super exciting for us. That's a growing enterprise. We also just got a set of funding from California Institute for Regenerative Medicine, CIRM, to become a part of the Good Manufacturing Practice, GMP facility network that was just announced. And that's an initial 2-year grant and then 5 years to follow, which is launching a 7-room manufacturing facility that's in the building next door. And that has consumed really a lot of my life over the last 18 months. So I'm very glad to have that finally launching. We're anticipating actually our California Department of Public Health licensing in December. And so that's going to be a huge milestone for us in terms of rolling along. We have some new faculty recruitments that are getting close to finalized. Be excited to be able to announce last year, and have those people come hopefully to this lecture series and be able to talk. All of that said, again, I just want to say thanks to everyone who made the time for participating in this series with us, and introduce Dr. Leslie Thompson, who is going to be telling us tonight, although we were just talking about it on the side, about Huntington's disease, which is more than just Huntington's disease. We were just chatting about how finding a more inclusive title is the way to go, because it's a great example of a neurological disease that gives us so many insights into how things can go wrong in neurodegeneration and, in fact, in CNS injury, and all of the ways that stem cells and genetics, in fact, stem cells in capital, genetics little, can help to tell us about those disease mechanisms and how we can think therapeutically from that perspective. Dr. Thompson is really preeminent in this field. She has some great stories. If you can get her talking, are you going to show any of that? About kind of the origins of the Huntington disease research consortium, which was, I think, really the first of its kind to bring together scientists from all kinds of different disciplines, not even just nationally, but globally, to tackle one single problem. There are a lot of research consortiums that have been launched subsequently on that theme, and she was really in on the ground floor of that. She, beyond being a really valued member of the Stem Cell Research Center here, she is the director for the Precision Health Institute for Precision Health. We had to change the order because of the acronym is awkward otherwise. Which is a really cool initiative that's aimed at using artificial intelligence and really breaking cutting edge technologies to apply to healthcare and ways to solve basic science problems and clinical problems and make things better. And that just reflects how much work Leslie does for everyone that is not just her and not just her laboratory on this campus. So she's a really wonderful colleague. And joining her this evening will be Dr. Devon Pendlebury, who is her fellow. Oftentimes we have these series where we pair a clinician and a scientist. Lately we've been breaking that mold a little bit and trying to pair someone who's an active researcher, a clinician or a PhD, with a student from their lab, a graduate student, a postdoc, a senior researcher who's really on the ground floor of trying to do all of these experiments that are just challenging to do and offering their perspective. So please welcome them, and thanks again for being here.
Hi, everyone. Thanks very much for being here. Thanks to the lab. It's a very supportive lab being here. So yeah, what we thought we would talk about is a project that Devon's been working on and give you a little bit of background about Huntington's disease and then why we're working on this project. And I'll try to bring in how this relates to other disorders and how we've been using stem cells to approach some of these problems as well. So it's titled DNA Repair as a Modifier of Huntington's Disease, and it is a new therapeutic target that's generating incredible excitement in the field, and not just in the Huntington's disease field, but any of the repeat disorders that exist, and I'll talk more about that. So what is Huntington's disease? This is where the genetics comes in. It is 100% genetic. It is inherited from either parent, so men and women get this disease equally. It's a dominant disorder, so if you carry that mutation that causes Huntington's, you will get the disease at some point in your life, or manifest the disease at some point in your life. And that means your children are now 50% at risk for inheriting this, it's really an awful disease. It strikes typically in the prime of life between the ages of 35 and 50, although there's juvenile forms of the disease where very young children can even manifest symptoms, or you can get it, I've known families where somebody's manifested disease at 80. So it's a real range of the lifespan. But typically, prime of life, you have young children, you're trying to build your career, and then you end up with Huntington's disease symptoms. And it's progressive. It can start very slowly, but then it progresses over about 15 to 20 years of symptoms before somebody succumbs to complications from the disease. So typically, someone will die, they will have a fall and hit their head, or they will have aspiration pneumonia. The symptoms of HD include a movement disorder. There's a very classic movement disorder that's involved. It looks through dance-like movements or chorea. But that's not what bothers people the most. The thing that's very difficult are the psychiatric and the cognitive features of the disease. So it involves what's called executive functioning. So it's the ability to make decisions, to recognize patterns, to be able to go, I'm going to put this load of laundry in the wash, and then I'm going to go cook dinner, and then I'm going to do this, and then I'm going to do that. And it's very hard. You lose that ability. You lose the ability to do your job. So we actually had a workshop this weekend, Devon and Charlie and others were at, where each workshop that we have for Huntington's will bring in a patient or a family and talk about how it's affected them, so that everyone's very motivated to share their very unpublished data and talk about how we can work towards solving this. And in both cases, both the young investigator meeting and the other meeting, the old person's meeting, they were two women who, if you looked at them, you wouldn't have seen any movement. You would never have guessed these two women had Huntington's disease. One of them lost their job in 2015. One of them lost their job in 2014 because they couldn't carry out those daily activities of life anymore. And that was devastating to them. They lost their sense of identity. In one case, their husband left. It's just a really impactful disease. And what it's characterized, so you can see, first of all, the little family tree here. I don't think we have a pointer, but excuse me, family tree here where you can track somebody that has the mutation, they call it gene positive, and track it across generations like that. And it is characterized with a particular region of the brain that's affected in Huntington's disease, and the type of cell, which will become important later on. It's called a medium spiny neuron. And this is an example of following autopsy, brain material from an individual that did not have Huntington's disease, was unaffected, and one that had died from Huntington's disease. And you can see the really profound loss, the atrophy of the brain, the shrinkage of the brain. But this region in here is very, very affected. So this is the basal ganglia, the caudate and putamen, and it should have this little butterfly shape where the CVS, the fluid runs through. But this is very enlarged in Huntington's disease, and that reflects the loss of the neurons, these medium spiny neurons in the brain. So that's the region that's profoundly affected, but what we really know now is that this affects many regions of the brain. And as Eileen mentioned, because this is completely genetic, but it also shares so many features with Alzheimer's, ALS, Parkinson's, many, many different neurodegenerative diseases, what we learn from HD can really be applied to many of these other disorders. So families are critical in any disease we study, any human disease. It's really tracking families that allows you to understand how a disease is caused, what are the characteristics of the disease, are there subgroups that are represented? This the sort of precision health hat is really understanding why we don't have any treatments for all these diseases, and part of why we don't have any treatments is because often they're not one disease. ALS is not, everyone with ALS is the same. It's even true for Huntington's, a purely genetic disease. Not everyone with Huntington's is the same, is going to respond the same way to a therapy. So we really need families to understand this. And for Huntington's, it's been critical in the identification of the disease gene that causes the disease. So this was first defined, this is back in the 70s, when this physician, Americo Negrete, had come to the US, gave a seminar on this Venezuela family, this large family or families who have Huntington's disease. And Nancy Wexler happened to be at that seminar. And she was trying to understand how we could find the gene that caused Huntington's disease. It turned out there's a large population of individuals around Lake Maracaibo in Venezuela. And so she went down there in 1979 with the idea at first just to see if there were any homozygotes. So this is a dominant disease. You only need one copy. That would be heterozygous for the disease. We wondered, so if you have two copies of the disease gene, is that worse? So she went down where people had, with HD, married other people with HD, and they transmitted both copies of the gene to their children and identified these homozygotes and found that no, they're no different than somebody that only has one copy of the gene. But what this allowed was this large pedigree of individuals to try to map the Huntington's disease gene. And this is just, this is Nancy Wexler, who is really my personal hero in life. And she would go down there each year along with Ann Young, shown here was the chair of neurology at Mass General. And they would go down and examine patients or people with HD, these families, track them, and get blood samples to really understand the cause of the disease. And so there was, through mapping, trying to find markers that would tend to be inherited along with HD to try to get fine-tune where the gene was on the chromosomes. And so as Eileen mentioned, this was a large collaborative research effort. This is Nancy here. At the very top up there is Francis Collins, who had been up till recently the director of the NIH. Also found the gene for cystic fibrosis. And a number of other individuals. I was quite a bit younger back then. And yeah, I know, all of us, right? Anyway, but it was really cool to be part of this. It took a long time. The first marker was identified in 1983, which you'd think would be a very quick process, but it was till 1993 that this gene was identified through these family studies and trying to track who had HD and who didn't from these large families. But one of the very cool things is, as Eileen mentioned, this was really a first of its kind in terms of having a consortia work on a scientific problem. And the authorship was the Huntington's Disease Collaborative Research Group. So it wasn't any one individual. It was 8 different labs, different people involved, but that was the author of the paper. And it's now been 30 years, which is hard to believe, since the gene was identified. And we don't have anything that changes the course of disease. At this point in time, there's nothing you can give a person that's going to influence when they get it, how long it lasts, anything about it. Only thing you can do is treat some of the symptoms. There's a approved drug to treat chorea. There's drugs to treat, so one aspect is psychiatric in nature, where there's a lot of aggression, there's anger, there's hostility, there can be psychosis, and there's some drugs to treat some of those symptoms, but nothing that changes the course of the disease, and which is just tragic, right? That we know so much about the genetics, but there's a lot we don't know. So that's kind of what we're going to be talking about today. So this is, the mutation itself is what's called a CAG repeat expansion. And so in the gene, the normal Huntington gene, we all have this gene everywhere in your body. It's expressed everywhere in your body. And it has this repeating CAG unit. When it goes up to about 35 repeats, you're not going to get Huntington's disease. 40 and above, you are going to get Huntington's disease. 35 to 39, it's not totally clear if you will or not. It's called incomplete penetrance. And this is part of a very long gene. It encodes a very long protein. It's called 350 kilodaltons. And when it's translated into the protein, it has, instead of these CAGs, it has an amino acid called glutamine, which is repeated over and over again. Again, 40 of those and above, you get the disease. Under 35, you don't. And that's illustrated. And then we know now there's a little teeny fragment that's very toxic that's also produced in the brain. And so again, if 27 to 35, those you will not get the disease. 36 to 39, it's reduced penetrance. 40 and above, you will get the disease. And above about 56 or so, that's when it gets into the childhood onset range for the disease. And in this range, though, you won't get it, but there is a possibility that your child will, that it could expand into the range, or right here, that it could expand into the range where you might pass along an expanded repeat. And this is where we are right now. You don't have to follow this in any way, shape, or form. But we've learned an awful lot about, even though we don't have a treatment yet, we've learned a lot about what that mutant protein, what that mutant gene does to a cell. And what it does is it affects pretty much everything in a cell, which makes it very complicated to know how to target it or what to do about treating the disease. And so what Devon and I are going to focus on here is something that happens to the DNA itself in the nucleus of the cell that seems to be really associated with how the disease is going to progress and when you're going to see symptoms of it and how people are approaching therapeutics. So one thing that people always ask is, well, why not just reduce the expression of that protein? Why not just get rid of it? So if you get rid of it, wouldn't you treat the disease? So it's been a huge effort over the last 10 years maybe to try to reduce Huntington itself. So it's called Huntington lowering. And there's many different ways that you can interfere with this. Sometimes it's at the level of the DNA itself or the message, the RNA that's produced that then produces the protein, and you can prevent that protein from getting made. And this looked like it was going to be very, very promising, and that it looked like Huntington could be lowered in CSF, in blood. But there were a lot of really disappointing trials. They were either stopped because they in some cases made people worse, or they just had no efficacy. And this was to target the total protein. So what I haven't mentioned yet is Huntington itself is absolutely required in the body and in the brain. It's very important for normal function. And so if you reduce total Huntington, you're reducing both the mutant one and you're reducing the normal one, which we need. So that, we've learned a lot from those trials though, a whole lot. And so there's quite a bit of hope now because there are new trials in progress that are targeting just the mutant allele, or there's ones that are using younger patients that are closer to age of onset where it might make a difference if you're targeting even total Huntington. And we've learned how you need to do clinical trials and the type of biomarkers you might use. So there's been a lot of really good news that's come out of this, but it was very, very disappointing to the families for obvious reasons. So what else do we know about this? Another aspect is that there is quite a bit of onset, a variation in the age of onset. So I mentioned that 40 and above, you get the disease. And so roughly, the longer the repeat, so at 120 repeats, somebody might get that at age 2. Or if you have 40 repeats, it might be around 50. And that's average age of onset corresponds to the repeat length. But there's a huge amount of variability in that. And that's especially true in the adult onset range between about 40 and 55 repeats. Somebody might have 42 repeats, get it as a teenager, or get it at age 80. There's a huge span of ages of onset depending on the individual. And what Nancy Wexler and the large team of the Venezuela Consortia found is that repeat length accounts for maybe 42 to 70% of the age of onset, but the rest of it is heritable. The rest of that variability seems to be genetic or genetics accounts for that other variability and somewhat environment as well. So what happened? What accounts for that? So what happened is the thought that if we could understand, not risk alleles, so when you're talking about Alzheimer's, you think of ApoE4 as a risk allele for getting Alzheimer's disease, right? You know that you have a higher probability of getting Alzheimer's disease if you have an ApoE4. In this case, they were looking for what might modify age of onset rather than what's going to cause the disease. We already know what causes the disease. And if we know that, could those be new therapeutic targets? So genome-wide association studies were done where you can look at the entire genome. You can look at all the DNA in an individual cell and figure out what genes might, with just a little variation, cause a difference in the age of onset. And these are, again, called genome-wide association studies. And this was the paper that we did. It was this 2015 paper where they identified pathways that were involved as modifiers. And the idea behind this then is that, let's say you have 45 repeats, what would make you get it later and what would make you get it earlier? So good modifiers, bad modifiers. It turns out most of these fell into the category of what are called DNA damage repair genes. So DNA, right, kind of a big deal, Nobel Prize, all that for the alpha helix. And we want to protect that DNA. We want to make sure that DNA is intact so it doesn't cause cancer, and likewise, it's not involved in causing neurodegenerative diseases. So what has been found is just really pretty landmark, actually, in our field, is so an individual's born with their CAG repeat. You're born with 45 repeats or whatever that is going to cause Huntington's disease. In this case, using the example with 40 repeats. So everybody starts with the same number of repeats, or an individual starts with the same number of repeats in all their cells. But in the brain, those can grow. Only in the brain. So in your skin cell, you're still going to, in your blood cell, you're going to still have 40 repeats. But in the brain, those repeats at a certain point in time in disease start to become longer and longer. And they will each have could have a different CAG repeat, and they can have very long repeats, up to even 1,000 repeats in the brain. And what's been found is this is pivotal, absolutely pivotal for Huntington's disease. So in the brain, you can have a mixture of repeats, so it's called somatic repeat instability for somatic tissue, not something that's going to go on to make an embryo or something, but it's in the brain itself. And so there's this new two-step model where you have over the course of a lifetime, slowly expanding these repeats in the brain, but at a certain point, there's a rapid expansion. And you combine that with all the toxic processes that we know happen from the presence of mutant Huntington, and you end up with the symptoms of Huntington's disease. And so what there's a huge effort now is to prevent that from happening, because if that is prevented, you don't transition to the really bad symptoms of Huntington's, if you can stop that from happening. And likewise, if you can prevent some of these toxic processes, you're going to be helping that process is all because they're all interrelated. All these aspects, DNA repair, other toxic effects in the cell are all interrelated. So one of the things we study in the lab is what's called SUMO, or small ubiquitin-like modifier, and this is a modification in HD. And you'll hear a lot more from Devon about this, but you got this little SUMO guy. It's attached to various proteins in the cell, one of which is Huntington itself. And this is a really rapid turnover process, so it's very hard to capture it and see it, but it's involved in everything in the cell. It's involved in proteins being cleared. It's involved in which proteins interact with who. You know, it's involved in DNA damage responses as well. And one of the proteins that we've been studying that came out of work from Joan Steffen and Jackie Geyer and Larry Marsh, who was here, is this protein called PIAS1, or protein inhibitor event activated STAT. And this enhances the SUMO modification of the Huntington protein and other proteins. And I'm not going to walk you through this, but there's a bunch of steps that go into attaching these SUMO moieties, or these SUMO molecules, onto a protein. And what's called an E3 ligase facilitates that. It sort of just helps attach the SUMO onto a protein. So we're targeting that in our studies with the idea that if we can prevent some of the things that are happening in Huntington's, we could help drive a therapeutic. We know that SUMO modified proteins, so proteins in the cell that have this molecule attached, accumulate. You can think of that as a buildup of garbage in the brain. And if we reduce the level of that ligase that attaches SUMO, we can reduce those smears. These are just high molecular weight smears on a gel. So it just reflects this accumulation of this modified protein. And so if we reduce the levels of that PIAS1, we can clear that, or we can prevent that from forming. And we found since then that if you can reduce the production of this, which is analogous to what we talked about lowering Huntington, but this case we're lowering PIAS, would this rescue some of the symptoms of HD? So Joseph Achaba, who was a graduate student a while ago, tested this in mice. He lowered the amount of PIAS1. You can see a wild-type mouse, with or without PIAS1, can scurry down a pole, so it's not bad for the mice. And then if you have an HD mouse, you can see it goes much, much slower down that pole. And then, so he takes a while. But if you treat him with lowering the levels of PIAS1, he scoots right down like a wild-type mouse, like a control mouse. So this suggested that maybe this was a good approach. And it's great that you can look at these mouse models, but we really want to look in the context of a human. And so we've, over the years used mouse models, fly models, yeast, C. elegans, worm models, but we want to look at humans. And so this breakthrough and I know everybody here at a stem cell center lecture knows this, but the ability to reprogram human skin cells, blood cells, et cetera, to what's called a pluripotent stem cell was a major breakthrough in 2008. And the premise behind this is that when you have a growing embryo, right, it's point of conception, those cells can then become different types of tissues. So, whoops. So they can become, they can go and become heart cells, skin cells, brain cells, etc. If you reprogram, say, a skin cell with various factors that brings it back to that embryonic state, that can now, again, differentiate to any cell type. You can do that now in a dish, which has offered huge opportunities for us. So again, you can take skin cells, reprogram them with a variety of factors to, again, this pluripotent state that can continually divide and can be guided to differentiate or form any kind of cell that you want to study. And Shinya Yamanaka won the Nobel Prize for this work, actually. But this has transformed our ability to research in Huntington's disease. Absolutely, we had no good cell systems to use, and this has really made a difference. And in the lab itself, there's a number of people that are doing different types of cells because we know that neurons are affected. We know that the blood-brain barrier is affected. We know astrocytes are affected. We know microglia are affected. So many of the people sitting in the audience are in those pictures, and they're all doing this work in the lab, and working also with another investigator here, Christopher Hughes, where they're making a little brain on a chip that you can even have artificial blood go through and understand the interactions between these different cells and study them in all sorts of contexts. You can bring these cells together, make co-cultures of an astrocyte and a neuron, HD astrocyte and neuron, and how they communicate with each other, and study the mechanisms that are involved. And we've really learned incredible amount over the years from studying this. And as well as now organoids, which are transforming brain research, which you can visualize them in a dish. This is just looking at them. You can see these little balls that reflect not brains, but sort of more complexity around what the brain would be doing in a dish. And so it reflects many of these cell-cell interactions that occur, so you can get at more mature, sort of older cells that can form in culture. But you can even see them. They're just really amazing. And Charlie's been leading this in the lab. And so we're using these as well to understand all the things that we're going to be talking about today. So we've done a lot of work in both mice and in these iPS cells. And this includes multiple mouse models where we find that we can increase their motor skills, so we increase their movement, their behavior. We reduce those smears you saw by knocking down pious. We know that you form the RNAs that are formed are corrected if you do that. And if we look in the iPS neurons and also organoids, we see that there's a lot of defects in these pathways that are rescued if you can knock down pious 1. And a lot of other types of phenotypes or symptoms in a dish that can be corrected by doing this. And recently a paper came out where there was a variant. So one of these small changes that I showed you in the DNA repair pathways in pious 1 that's associated with a later age of onset. So it's even come out as a modifier of age of onset. And so we have a lot of efforts going on now to develop therapeutics. I'm working with Ionis to develop an ASO, small molecules with our therapeutics center on campus, viral delivery as well for knocking this down. So I'm going to hand it over to Devon, who's going to explain some of the interactions between these different pathways and how we're studying it in the lab.
Thank you. All right, so hi, I'm Devon, and I'm going to continue to talk about DNA damage in Huntington's disease. So there are many hallmarks of neurodegenerative diseases, such as pathological protein aggregation. Each neurodegenerative disease tends to have a characteristic protein which causes aggregates associated with it, neuronal cell death, and also DNA defects, including DNA damage. So it's something that's important not just in HD, but also across many other diseases. And so we can see that DNA damage accumulates in Huntington's disease mouse models as well as patient brains as compared to unaffected patients. So here I'm going to show you mouse models. So H2AX, which is the protein shown in green here, is a DNA damage marker. You can see in this mouse model at 4 weeks and 12 weeks, there's drastically more DNA damage in the HD mice versus the control mice. And the red protein here is just showing us where the neuron nucleus is, so we can see where the DNA is located. And we can see the same phenotype if we look in postmortem human tissue. So there's just a greater amount of DNA damage in HD patients versus unaffected patients. And what are the causes of this DNA damage in neurons? So in other cells in the body, when cells undergo replication, that's a major cause of DNA damage. But in neurons, they're no longer dividing, so that's not a cause here. Some of the causes can be transcription. When our genes are read to try and produce the proteins, sometimes you have to manipulate the DNA to unwind it so that the genes can be read properly, and this can create DNA damage. Metabolic stress, neurons are highly active, and metabolism can cause reactive oxygen species, which can cause damage to DNA. There's defects in protein turnover in diseases, and this can affect proteins which are important in DNA damage repair, as well as protein aggregation. Proteins which aggregate in diseases including HD, such as the Huntington protein, a lot of them are actually involved in DNA damage repair themselves, so this can also affect DNA damage. And there are different flavors of DNA damage, you could say. Sometimes you get a mutation, which is where one nucleotide in the DNA is switched out for another one. And sometimes this can cause mutations in the protein which is made from this gene, which can have negative effects. Nucleotides or bases can get modified. Some chemicals in the body can attach themselves to a base, and this can have damaging effects. You can get a single-strand break where one part of the phosphate backbone is broken, or you can get a double-strand break where both backbones are broken. And I'm going to talk about double-strand breaks in HD today. So the good news is that our cells have evolved many, many ways to fix DNA damage. Our cells encounter thousands of DNA damage events throughout the day. And there's, as you can see here, several different pathways which they can use to fix this depending on the kind of damage. So I just want to point out here, and Leslie was talking about proteins, DNA damage repair proteins involved in modifying the age of onset of HD. These are proteins which are involved in a mismatch repair pathway on the right there. What I'm going to talk to you about today is the non-homologous end joining pathway. So that's a way of repairing double-strand breaks, and it's a pathway, and it's the Huntington protein itself is involved in this pathway. And so here I'm showing you the wild-type Huntington protein, and it's part of what's called a transcription-coupled non-homologous end joining, or NHEJ complex. And so transcription is the process of DNA being turned into RNA. So DNA is read by an enzyme called RNA polymerase, and this creates an RNA transcript, which is then read and turned into protein. So this Huntington is part of a transcription-coupled complex, meaning that the RNA polymerase itself recruits Huntington and these other DNA damage repair proteins to sites of DNA damage that it finds during transcription when it's reading the gene. And this allows damage to be repaired quickly and efficiently so that the polymerase can carry on its business. And so Huntington is a very big protein and can act kind of as a scaffold to bring lots of different proteins into this complex. And so there's many different NHEJ DNA damage repair proteins in this complex, and they all get recruited, fix the DNA damage, and then everything is again. But what happens when in HD, mutant Huntington is expressed in cells instead of the wild-type Huntington protein? So as Leslie mentioned, the mutant Huntington has an expanded polyQ repeat sequence. And we also, like I've shown you, there's a increase in DNA damage and decrease in DNA damage repair in cells with mutant Huntington. And so kind of our hypothesis and something we're looking into in the lab is that mutant Huntington, when it gets incorporated into this DNA damage repair complex, disrupts its extended polyQ repeat, kind of changes the structure and the behavior of the protein. And it's disrupting these recruitment of these key DNA damage proteins, and it's affecting their function. And so I'm going to kind of walk through an experiment for how proteins in the Huntington DNA damage repair complex can be identified. And so an experiment was done by our collaborator, Postdoctoral. So we started with just a control mouse, and we isolated the nuclear proteins from a mouse brain. And so in the nucleus, there's lots of proteins, some of which are involved in this Huntington complex, some of which are not. And then we can incubate with an antibody, which binds specifically to the Huntington protein. So antibodies are found naturally in our bodies. They recognize certain specific parts of proteins. So you can get good antibodies, you can make them, and we use them a lot in the lab, that will bind a specific protein. So we add one that will bind specifically to the Huntington protein, and we can kind of pull out of that complex anything that Huntington protein and any other proteins that are bound to it. And then we wash away anything that's not bound to these proteins. And we can analyze what's there either by using a Western blot or mass spectrometry. So I'm going to show you a Western blot. So in this case, I would kind of run this protein on a gel, and I'd use a different antibody to look if that protein is part of this complex or not. So here's an example of what that looks like. So on the left here, these are the input samples. So all these proteins were there when we started at the beginning, before we pulled down with the Huntington antibody. And on the right here are our IP samples. And on the right, these are all the protein antibodies that we're probing with. And so if you see a band show up here, that means that protein is there in our pull-down sample with the Huntington protein. And I want to point, and so this is not a complete list of proteins that are in this complex. There's lots of other ones, and kind of we don't really fully know the changes that are involved in this complex in wild type versus HD cells yet. But what I want to point out is four of these proteins in this complex, Huntington, TDP43, FUS, and ataxin-2, those are all major players in various different neurodegenerative diseases. So TDP43 mutations can cause ALS and FTLD, as well as FUS and ataxin-2 plays a role in ataxias and also ALS and FTLD. So this complex could have implications not just in HD, but also in other neurodegenerative diseases. All right. So as Leslie mentioned, something that our lab studies a lot is the sumo modification of proteins. And it's very well established that DNA damage repair proteins can be sumolated. And this sumolation can have various different effects depending on the protein that it gets attached to. So in some situations, a sumo attaching to a protein can encourage it to bind to other proteins. This kind of can help sometimes recruit proteins to certain areas in the cell. Conversely, sometimes the addition of sumo can totally disrupt protein interactions. It can do the exact opposite, and it all depends on when this is happening and the protein it's going to attach to. Similarly, it can detach a protein which is bound to DNA during DNA damage repair, or it can help recruit proteins to sites of DNA damage. So this is actually very confusing to figure out how sumolation acts in DNA damage repair. But kind of the bottom line I want to get across to you that it's just a balancing act. The assimilation of proteins needs to happen in a certain order. Proteins need to be recruited to DNA damage. They need to do their job, and then they need to leave. And if, like we see in HD, where we get that excess of sumolated proteins, if the balance is disrupted, these pathways can stop functioning efficiently. And so I want to talk about a specific example of this from our lab. So PNKP is a DNA damage repair enzyme. Its job is to kind of process the broken ends of DNA so that they can then be repaired. And our lab has shown that if PIAS1, which is the sumo ligase Leslie was talking about, if that facilitates the stimulation of PNKP, it decreases PNKP's activity. And so this is fine in normal cells where it's part of that kind of stimulation-destimulation balance. But in HD, where there's an excessive stimulation, it leads to an a decrease of PNKP's activity and therefore an increase in DNA damage. And one way we can look at PNKP activity is to use this assay. So I can't reach up there. But there's a radio-labeled DNA end at the DNA damage break. And if PNKP is working and it's active, that radioactive phosphate gets removed, and we can see that on a gel. And if PNKP is not working, we'll see less of that. And so here's an example of how that looks. And so this is the phosphate that's being released on the blood. And so in wild type, we see this much, and you can see there's less in the HD sample. But if we decrease the levels of PIAS1 in the cell, you can see in wild type, we see an increase in that released phosphate. And in HD, we also see an increase. So again, kind of reiterating what Leslie was talking about, PAIS1 is a great target in HD, and it can affect DNA damage repair. Yes, and you can do this in iPS cells. Okay, so something I've done a lot in the lab since I joined is to determine which DNA damage repair proteins can be sumo-modified. And how we do this, we use HeLa cells. It's a cancer cell line, and cancer cells are known to have high levels of sumylation. So that's why we use that line for this particular assay. We transfect in a his-tagged sumo1 or sumo2 protein and a MYC-tagged protein of interest. So those tags are just there to help us identify the proteins later on in our experiment. So the cells will make those proteins, and if a protein is capable of being sumo-modified, that will happen in the cell. And then we use beads which bind to the his tag on the sumo. So anything that's got sumo on it will bind to these beads, and we can wash away anything else. And then we can do another Western blot to look for a size change. So if we get a sumo added, we should see a shift in the molecular weight of that protein. And so this is just an example of what that looks like. So here's a protein without sumo, and then when you add sumo, you see an increase in the size. So anything towards the top of the gel has a bigger size. So you can see that all these proteins on this blot have been sumo-modified. So many of the TC and HEJ complex proteins can be sumo-modified. So that's kind of something that we're actively studying in the lab, is how, if there's an excess sumylation of these proteins in HD cells versus control cells, and if this is affecting DNA damage repair, like in the PNKP example that I showed you. And so the kind of the next steps are to use patient-derived iPSC neurons and differentiate them to patient-derived iPS cells and differentiate them to medium spidey neurons and compare the composition of this Huntington DNA damage repair complex and see if there's differences in HD versus wild type, and kind of try to figure out what's different in HD and if we can target that to increase DNA damage repair. Okay. So something else that I'm actively interested in the lab, and so are many other people. As I mentioned earlier, TDP43 is a part of this DNA damage repair complex with Huntington. And TDP43 is a huge player in many neurodegenerative diseases, particularly ALS and FTLD. And it's an RNA and DNA binding protein. So this is just a pretty structure here of TDP43 bound to RNA. And it plays roles in RNA metabolism, splicing regulation, and gene regulation. And so here, just TDP43 is in our complex. And so in a healthy cell, TDP43 is primarily located in the nucleus of this neuron. And as you progress towards neurodegenerative diseases, you see more of a cytoplasmic accumulation of TDP43, and then you slowly start to see it move away from the nucleus and aggregate in the cytoplasm. And this is a huge hallmark of ALS, and now we're also seeing a similar phenotype in HD. So Ty and Alice in the lab took these images from HD patient brains. And you can see, if you look at TDP43, and the phosphorylated TDP43 is found in disease, you can see it outside of the nucleus. So the DAPI, the blue here, is where the nucleus is, and you can see that TDP43 is being found outside of the nucleus, which should not happen in a healthy cell. Additionally, since we love to look at simulation, we can also see TDP43 can be sumo-modified. It's actually super interesting because TDP43 has many post-translational modifications, and phosphorylation and ubiquitination are highly relevant in disease, especially ALS. But simulation is not as well studied. So we're something I'm kind of looking at here is how simulation will affect TDP43's DNA damage repair function. And also how simulation can affect TDP43 mislocalization in the cell, and whether modifying simulation can kind of get TDP43 back into the nucleus in some way. So kind of the final message I want to leave you with here is that DNA damage repair, both the DNA double-strand break pathway that I was talking about and the mismatch repair pathway that Leslie was talking about, are both targets that we can study, and we can try and identify new therapeutic targets to try and improve HD, try and improve the disease progression and the outlook for HD patients. With that, we would like to thank the Thompson Lab. Thank you all for staying late to come, as well as our funding and the Stem Cell Center.
And just to tie a few things together, so this is an example of how you have to sort of dig way deep into a particular area. In this case, this DNA damage repair. We're using stem cells also, many of you have heard, excuse me, for transplantation in HD brain as a way to also repair the cells that are there and keep them healthy. So there's many different ways that we're using stem cells, including what you just saw, which is an unbelievably exciting area of research for Huntington's. So again, thank you very much.
All right. Don't put that down. So thank you both. Now's the part where they get to harangue you.
No problem.
I have one quick question, maybe, because we have a number of people in the audience who are, you guys, of course, met with beforehand, that are coming from the perspective of sort of understanding research better, right, and why we do some of the things the way that we do. And one of the problems that we can have in research is, for lack of better words, coming to me at whatever, 8 o'clock at night after a long day, is a chicken and egg effect, right? And so it seems to me, it's really cool data, right? But how do you pick apart the chicken and the egg, right? What is causal here versus what's just messed up by virtue of having these wacky expansions that are taking up space and stuff in the cell?
That is an excellent question. If I knew the answer to that, I would have a company and I would, no, but, no, but so the mutation happens. We know that there's effects at the DNA level, the RNA level, the protein level. Messes up the cell. We know it messes up DNA damage repair, possibly through, as Devon mentioned, even the clearance of those proteins is messed up, so you're going to mess up the DNA damage repair. It does seem that at least one pivotal thing in HD that we've found over the last couple of years, we being global we, is the fact that you have that somatic repeat. So we've known for years that that happens in the brain, but didn't know until these modifier studies that that was pivotal to how the disease was going to progress or when you're going to show symptoms. So it seems to be a causal effect that's caused by the repeat expansion being there. So still can't answer exactly chicken and egg, but that if you can, at least we know where you might want to intervene finally. And that's huge.
I'm going to be so rude and ask a
You
second
can
question.
totally
but
be rude.
So we do this faculty meeting thing once a month, and Leslie gave the first one a long time ago. It's like a proposal development meeting.
Yeah.
And it's terrible to put a bunch of faculty in a room together because we all just start asking one another
Except it's
questions
great
like they're
because it's
graduate
better
students.
than any review process.
So there are other trinucleotide repeat diseases, right? That
Yep.
aren't CAG expansions. They're expansions of other stuff. Do they cause these same things?
Yes, so that is what's coming out of this is super interesting. So a certain form of ALS that's caused by a hexanucleotide repeat also seems to be involving some of the DNA damage repair proteins. It's a very new area, like only a couple labs working on it. The CAG repeat expansion diseases, which there's like 10 at this point, all of them, it's starting to show that this is involved, including including TDP43. But one that was super interesting, so one of the big questions we went through in this workshop we just had is how in the world are we going to use this knowledge to treat the disease, because you interfere with DNA damage repair pathways, you can cause cancer. So you might want to try to get it into the brain, maybe not in the periphery. We don't know what levels, like some of these proteins you can get rid of and it's fine. An animal's not going to get cancer. Some of them, it's really bad. So we have to understand that. And then, because the repeat expansion only happens in the brain, how are you going to test if that drug is working before somebody passes away from the disease? You can't get a biopsy of the brain. So we need markers in the blood to tell us that you've interfered in some way. But so what was cool is that, so this one disease called SBMA that Alice Bada here at UCI works on affects both muscle and the brain, and both in muscle and the brain you get this repeat expansion. So if you get in the muscle, you can do a muscle biopsy. So that may be one of the first diseases that could get tested. So it's just exploded in the field right now.
I work with developing a chemical for prions. Is there any kind of correlation between prions and Huntington disease?
Yes, so not in this pathway per se, but the whole field of protein aggregation diseases, prions, in Alzheimer's you have A beta, you have tau, you have, all these diseases have protein aggregation issues. And even some of the transmissibility seems to happen in all these diseases, way more in prion diseases, right, where it seeds itself, it goes from cell to cell to cell. You also have some of this cell-to-cell transmission in Huntington's and Alzheimer's and other diseases.
Great talk. I just was thinking about, is there also like somatic expansion in other diseases as well? Like where you...
Of like CAG, there is in the other CAG repeat diseases, like the ataxias. I don't know if there is in the C907 too. I think, yeah.
We think there is in C9.
I think if any repeat gets to a certain length, then
Fragile
it's
X,
very unstable.
myotonic dystrophy, all of those, Friedreich's ataxia, it looks like all of them have some kind of repeat instability in some tissue,
Yeah.
especially the brain. Many of them, so the MSH3, so one of them that's been modified the most, or perturbed the most, has been implicated in things like Fragile X and myotonic and other disorders. So it's just really emerging.
Sorry. Just having a little silent conversation.
So in theory, Huntington is in every cell in the body.
Not in theory,
No,
it is
in-
there.
Yeah.
Only in theory, because only the spiny neuron.
No, so, okay.
Why?
No.
So those are the ones that you can tell die, that they're gone. But there's dysfunction all over the brain. So if you're looking at the functions of the other cells, those are also impacted and really early in disease. And we know pancreas is impacted, heart is impacted, gut is impacted. Many different cell types throughout the body are actually impacted in HD. It's just that we focus so much on the striatum because they're gone. It's the first thing you see.
So you could do that a liver biopsy.
But we don't know if there's not repeat instability there. You could do but you don't. So you have dysfunctional cells, but you don't necessarily have the huge amount of repeat instability you see in the striatum. So, and we're finding with other diseases, I don't need to go into this, I guess. But anyway, there's some really interesting science in other diseases in the cell types that have instability.
I'll ask later.
Sorry, I'm just a little bit confused. So for people with Huntington's disease, what makes it different so that their CAG repeat expands with somatic expansion over time versus regular people?
Do you want to take a stab at that? That's no confusion. That's a great question.
It only CAG repeat gets to a certain length, and then you see the expansion, in people without HD, it's not long enough to have that effect. It's only when you get to a certain length. And then the longer it gets, the worse the expansion gets. And after a certain point, it gets really fast. Yeah, and then some people also have mutations in the modifier genes, which either increase or decrease that repair pathway that facilitates expansion.
So also, one of the areas of research right now that was like the first half of this workshop was to get at the nitty-gritty. What is different about that DNA? What is different about the structure that is it during replication of the strands of DNA that you have this happen? Is it because there's a small loop versus a big loop, and so then it doesn't get repaired properly? I mean, there's just, like, there's this really basic science going on to understand exactly your question
not
of
fully understood yet.
why it expands.
Who else?
This is kind of off topic, but do you see any future for the cyanobacteria dealing with these neurodegenerative diseases?
I'm not totally sure how they've been using them, but it's entirely possible. I'm not as familiar with that area,
Okay.
but I'd love to hear more about it. Yeah.
So in the brain, there is active DNA replication in there?
So
Thank you.
that is an excellent question, and we didn't clarify that. I don't know if you want, I don't want to answer all. Okay. So no, there is not, except for some of the, there's what's called neurogenesis. So there are some new neurons or new astrocytes that get made, new cells that get made in the brain, but in general, they're non-proliferating, they don't have DNA replication. But they do have DNA repair happening. So when we were talking about this repeat expansion, that's as this whole as it's developing, right? As you go through development, there's proliferating cells and DNA replication, and that's, you know, when you're passing on, say, during spermatogenesis, there's a lot of DNA replication, there's a lot of repeat instability in sperm. One of the things that was collected in Venezuela actually were sperm samples to study that exact thing and to see, does age make a difference, how many repeats you pass on? Because juveniles always from the father. It seems to be the instability in the sperm. And so, you know, is age a factor? Is you know, what causes the repeat instability? When do you get these huge expansions and why? So you're absolutely correct, there's not replication in the neurons, but yeah.
So I was just going to clarify, you missed, so it's transcription coupled repair that can, so it's when genes are transcribed, it's not when DNA is replicated that this can happen. So.
Yeah, for the, but also mismatch repair, right?
Yeah.
So at the DNA itself, so during, and one of the things that was found was actually the somatic repeat instability requires active transcription to happen. So that's another factor.
So, great talk. I'm really interested in if simulation's involved in the protein dissociation or kind of association behavior, if you know if affecting simulation will change protein aggregation behavior in a negative way.
Oh, I think a lot of the times adding sumo onto a protein makes it more soluble, which kind of is both good and bad. It's kind of good in that a protein is not necessarily aggregating then, but sometimes with mutant proteins such as Huntington or something that needs to be cleared, the solubility can prevent it aggregating, which then prevents mechanisms in the cell from clearing the aggregation. So it can be both good and bad. But it can probably work both ways. It tends to have very different effects depending on the protein it's attached to.
And I think you're referring also to those smears, right? So the whole, everything is messed up. So protein clearance pathways, as you well know, are impacted, right? So you're not clearing these sumo-related proteins effectively. They're also modified by tons of other things, and so they start glomming up together, and we don't know if those are actually bad or good in
Yeah.
Huntington's, right? They may be a storehouse to get rid of toxic protein too. So it's not clear.
Audience, anybody else? Students?
This guy.
You are a faculty
Is the primary damage to motor function from Huntington's due to like mass apoptosis or literal dysfunction in the cells?
Actually,
Both.
there's not widespread apoptosis in the brain in Huntington's. It's more dysfunction and different types. Like, we don't actually know what causes the cell death in the brain because you don't see markers of apoptosis, but it could be other forms of cell death. But there's massive dysfunction.
So I was going to ask Devon, you don't sound like you have a Valley Girl accent. So I'm wondering if you could tell us what you did as a graduate student. How did you end up as a postdoc here? So why are you doing what you're doing now, and then where are you going with that next?
I'm still British.
No, there's an infectious protein in the ocean that you've been transformed.
So I did my undergraduate in the UK, and during that time, I took a year out to do research, which is when I first came to the US. So I worked at the Mayo Clinic in Jacksonville, Florida for a year. I did research. It was a great experience. And then I decided to come back and for several reasons and do my PhD in the US. So I did that at the University of Michigan. I see there's an Ohio State fan in the audience, which upsets me. And I am actually I did my PhD in structural biology. I love to look at what proteins look like. I love to stare at individual amino acids and see what they're doing, what happens when you mutate them, that kind of thing. And so I've always been interested in neurological diseases because of the protein aggregation, the changes in protein structure and function. It's really a protein-driven disease.
And next?
Yes. So when I graduated, I came to UCI. I was initially started in Rob Spitali's lab, and then he closed his lab. And then I ended up, fortunately for the better, I met Leslie, and she has a great passion for HD research and trying to find a cure. And now I'm part of the HD community, and I really love working. And so I'm a postdoc now, so I research, and I do hope to eventually go on and run my own lab.
And she's actually very involved in the screening for a compound through the Neurotherapeutic Center with all her chemistry and structural biology expertise to find something that binds to pious and can reduce its activity.
Any last questions before we close up? Oh no, you're getting trumped.
Sorry. Throughout this entire talk, one question that has been really on my brain is, why 39? Is there something special about 39 net repetitions being the cutoff for it becoming either a silent gene that later gets passed on possibly, or a dominant gene that for sure is going to get passed on? You're right.
So it just seems to be a structural threshold. So both a structural at the DNA level and at the protein level, where the kinetic, the rate at which it can aggregate increases profoundly, or the rate at which you can get DNA damage repair defects increases, because you get these structures that form at the DNA level. There was one of my favorite experiments of all time in HD research was a worm model where they took these polyglutamines and they did it sequentially. So they showed it's very soluble, very soluble, very soluble, and they hit 39 and 40, and there was this abrupt transition where you then saw it aggregate. It was just amazing. It was absolutely black and white, or green and not green.
So that's actually what I was going to kind of jump in and ask before we hand it on for one last closing amazing question. And so it's a flip that allows the protein to aggregate, as opposed to if you had 38s but you lived to 150. Are you going to end up with?
So 35 to 39 is incomplete
Okay, if
penetrance.
you had 25.
Okay, if you had
yeah, and
30,
lived to
let's say, yeah.
200, right?
Yeah. You wouldn't get it. Would not get it. That there is an absolute cut, like 35, 34 and below, you're not going to get. We all have that gene in our body, all of us. I probably maybe have 18 repeats or 20 repeats of mine. You know, it varies anywhere from 14 or so up to like 21, well, not 14, maybe 18 to 25 repeats, something like that typically. And you start getting these repeat expansions, which people are studying how that happens. And then it flips a switch.
All right, closer.
That was a lot of pressure. Yeah, lots of pressure. So with all the interest in making PIAS-1 knock down a therapeutic, I'm curious if the level of simulation has been looked at in an age-related manner, and if it has been compared to the level of protein aggregation that occurs in Huntington.
Not yet, because it's, as I mentioned, a very transient process. And for the first time, because of something that's gone on in the lab, a research area, we can actually capture those proteins now. So we have not been able to do that. And should mention that Nikki over there, she's also doing all these different cell types, differentiating from iPS cells into heart cells and gut cells and brain cells and astrocytes and everything to see what is different in those cell types.
I like how you closed on the power of stem cell research, so we'll hold it there. If you guys could help me thank our speakers for this evening, and once again, thanks for coming.